I'd like to thank the organizers for the opportunity to speak. Over the next few minutes, I'll talk about messenger RNA and lipid nanoparticles for the induction of HIV protective antibodies. The hypothesis of the Duke Consortium for HIV/AIDS vaccine development, a team, is the induction of high-titered, durable polyclonal serum and mucosal broad neutralizing antibody responses to multiple HIV envelope epitopes will be protective against HIV transmission. The goal for our team over the next 4 years is to achieve proof of concept that a broad neutralizing antibody vaccine can be made. The criteria for this proof of concept is that we want to induce at least one type of broad neutralizing antibody to achieve greater than or equal to 50% serum breadth against hard to neutralize viruses called R5 tier 2 transmitted founder viruses with antibodies that bind to a broad neutralizing epitope in monkeys or humans, preferably humans. Secondly, to induce serum and mucosal broad neutralizing antibody levels for a minimum of a year in monkeys or humans. The challenges of eliciting HIV broadly neutralizing antibodies are, number one, that the envelope is the target for neutralizing antibodies and its envelope is covered in sugars shielding the HIV glycoprotein from the immune system. Secondly, immune tolerance can be a hindrance. Broadly neutralizing antibodies can be disfavored by the immune system thinking that broad neutralizing antibodies should not be made. Third, broad neutralizing antibodies have high levels of mutations or changes that occur over time in its sequence, and many of these critical changes that turns antibodies into broadly neutralizing antibodies rarely occur. And Kevin Saunders will talk about this problem later on in this session. And fourth, easily made envelope antibodies are not broadly neutralizing and can divert the immune system, such that non-neutralizing spots in the HIV envelope can induce antibodies that outcompete rare broad neutralizing antibody precursors. This slide shows the strategy that many in the field are using to overcome the disfavored status of broadly neutralizing antibodies by designing and developing envelope immunogens that can stimulate the precursor B cells, the naive B cell receptors of broad neutralizing antibody lineages, and then coming in with sequential immunogens that can expand these lineages and then select for B cell receptors that are affinity matured and have acquired all the changes or improbable mutations that are needed for mature broadly neutralizing antibodies. Today, I'll just talk about some progress made in inducing CD4 binding site and some progress made in inducing GP41 membrane proximal external region targeted broadly neutralizing antibodies. The rest of these and this list here are other types of antibodies that we and others are trying to induce. The focus of the talk today is the platform nucleoside modified messenger RNA and ionizable lipid nanoparticles, or mRNA LNPs. Of course, we all know that 2 COVID-19 mRNA vaccines have been approved by the FDA. They are safe. There are no viral components other than the immunogen itself encoded as mRNA. There's no risk of genome integration. Very high immunogenicity, not only for COVID, but also for Zika and influenza have been shown, and ease of production, and that one can rapidly and cost-effectively do large-scale manufacturings of mRNAs over a shorter period of time than for proteins. Now, the ability of the mRNA to be a good vaccine depends upon the lipid nanoparticle to release the mRNA on the inside or the cytoplasm of the cell. An mRNA is released from lipid nanoparticles and is translated in the cytoplasm, and that means that the protein that the mRNA encodes is produced and is either secreted or put into the membrane of the cell and then is recognized by the immune system as a vaccine. Now, another roadblock is that the titers to prevent HIV infection are high. The serum inhibitory dilution 50 titers needed to achieve 90% protection in a meta-analysis of non-human primate studies has shown that for CD4 binding site antibodies, it's around 1 to 500. And also for broad neutralizing antibodies that target the first and second variable region, it's also about 1 to 500. Interestingly, membrane proximal external region broadly neutralizing antibodies appear to be more potent and only require a titer of about 1 to 15. Other issues for encoding a complex trimer immunogen with messenger RNAs are that broadly neutralizing immunogens must be well folded to express broad neutralizing epitopes and to not express the diverting non-neutralizing epitopes. And then secondly, that means we must define stabilization strategies of well-folded envelopes that result in the majority of messenger RNA encoded molecules being well-folded. We've done this in a paper recently published by our team in Cell Reports this year, in which we reported that mRNA LNP can encode for multiple complex forms of HIV vaccines that can be made by messenger RNAs, including trimers, envelope nanoparticles, and cell surface ONVs. We also showed that mRNA encoded HIV on trimer ferritin nanoparticles can induce monoclonal antibodies that neutralize heterologous HIF1 isolates in humanized mice. So the first specific epitope that induces antibodies is called CD4 mimic classes of CD4 binding site broad neutralizing antibodies. These are two types of antibodies. One is called the BRCA1 class. The other is called the 8ANC131 CH235 class. They're similar in breadth and very potent, and they bind to the HIV envelope in a manner similar to the CD4 at the CD4 receptor binding site. One of the antibodies called CH231 uses many different light chains, whereas the BRCA1 class uses a very rare light chain. And the key is to design immunogens that can initiate and complete the development of these antibodies to full neutralization potential. We've initiated such a lineage, a CH235 type of lineage, by the following criteria, and that vaccine-induced CD4 binding site broad neutralizing precursors in macaques have a signature binding mode of binding to the ONV in the same orientation as CD4 itself binds. On the left-hand side, we see a cryo-EM structure by Priyambada Acharya and her team with the trimer in light gray and the GP of 120 and the GP41 in black, and the antibody in light and dark blue binding on two sides of the trimer as shown here. And if one looks closer, one can see that the antibody has a signature type of binding to the CD4 binding loop and the CD4 binding site by binding with a particular part of the antibody called the HCDR2, which is highly similar to what CD4 does itself. So we know that we have induced these kinds of antibody lineages, and the goal now is to boost them with boosting immunogens to lead them to mature. So the status of CD4 binding site immunogens is that we have a nanoparticle protein manufactured, and clinical trials should begin the first of the year 2023 with this protein. And the boost has already been produced GMP that we know will select for more changes in the antibodies, more rare changes in the antibodies to move them towards broadly neutralizing breadth. With regard to mRNAs, primes and boost of mRNAs as GP160s for these same immunogens have been accomplished, and these will be compared with proteins in clinical trials. And the final boost are being developed in the CHOB-D by the CHOB-D team led by Kevin Saunders and Kevin Weehee at Duke. Now, the last target that I will show is the membrane proximal external region that is a broad neutralizing epitope down at the base of the trimer, right next to the viral membrane as shown here. We've just completed a clinical trial with a liposome in which is attached a protein T-cell helper epitope, to which is attached the broad neutralizing epitope shown here and as the blue line. This has been studied in monkeys where we initiated a broad neutralizing antibody lineage and got it to mature to the point of very weak neutralization. The same immunogen, interestingly, in humans induced B-cell lineages to acquire heterologous neutralization breadth, and we're undergoing studies to determine the extent of the breadth of the heterologous neutralizing antibody B-cell clone. So we're excited that we have moved the B-cell lineage from the precursor stage to heterologous breadth in humans. So the status of Emperor immunogens is that the Emperor peptide liposome immunogen has induced proximal GP41, Emperor BNAP precursors, and also induced mature neutralizing antibody clonal lineages in humans in HVTN 133 clinical trial, HVTN being the HIV Vaccine Trials Network. The prime and boost for both proximal and distal, the two different epitopes in the membrane proximal external region, are in preclinical development as mRNAs to induce both of these with a single immunogen. The way we're doing this faster is that we have our own good manufacturing practice, vaccine production facility and capability at Duke for the CHOV-D work. The suites are designed with flexible single-use systems and ability to operate independently, so we can make more than one process in each of the 5 suites. The timelines for mRNA LNP are being accelerated in order to be able to perform iterative clinical trials very rapidly in the HVTN. Preclinical down selection has been decreased to 10 weeks, and this is in collaboration with Drew Weissman's lab at the University of Pennsylvania. GMP manufacturing has now been truncated to be totally about 5 months from start to finish. Dedicated regulatory process development analytics and manufacture teams are now in place for each product. We have 2 full-time teams, a total of 12 individuals for the CHOV-D products dedicated solely to this work. And we aim to have overlapping product development such that 2 to 3 mRNAs per year can be tested in the HIV Vaccine Trials Network experimental medicine phase 1 trials. So in summary, we're making progress on inducing proximal Emperor and CD4 mimic CD4 binding site BNAB B-cell lineages. The goal is to induce both lineages in humans to full broadly neutralizing antibody breadth using mRNA LNP by approximately 2025, with the ultimate goal to combine immunogens for a bivalent sequential vaccine for an efficacy trial of a prototype HIV vaccine, again in the HVTN. We'd like to acknowledge our funders, primarily the National Institutes of Health Division of AIDS, National Institute of Allergy and Infectious Disease, as well as the Bill and Melinda Gates Foundation, and all of our partners, in particular the HVTN for clinical trials. For the preclinical team, it takes a village to do this kind of work, and in particular acknowledge Drew Weissman and his team at the University of Pennsylvania as our colleagues and collaborators for the messenger RNA lipid nanoparticle work, as well as Acuitas for their lipid nanoparticle support. And acknowledge our clinical trials team in partnership with the HVTN. Thank you very much.